Nucleic acid. Okay. So you might have heard of nucleic acids. Types of nucleic acids. DNA and RNA. So what is DNA? Deoxyribonucleic acid. This one is ribonucleic acid. So DNA, RNA deals with what? What kind of in the context of DNA, RNA?
Cell genes.
Heredity. Genes.
So it talks about heredity. So heredity is basically where we have transmission of inherent character. So from parent to child, or that basically you have something known as chromosomes, right? So chromosomes you will have proteins and nucleic acid. So chromosomes heredity varo. So chromosomes you will have proteins, which we studied, and you will also have nucleic acid, right? So what is nucleic acid basically? Nucleic acid is, we call it as a long chain polymer of nucleotides. Nucleotides. So polymer. So we studied first polysaccharides. It was a polymer. Then we studied proteins. Proteins another. So polymer of what? Polymer of
Alpha amino acids.
Alpha amino acids. We say that nucleic acid is nothing but a long chain polymer of nucleotides, or we generally call it as, and it is called as polynucleotides. So when I say polynucleotides, it is nothing but nucleic acid. Okay. Sorry. So what is nucleotide? So first we need to understand first what is nucleotide and how they are linked together. Because when I say poly, it is linked together. So we need to understand what kind of a bond it is there. So we know that polysaccharides, monosaccharides join together to form polysaccharides. In a bond, what was that bond called? Glycosidic linkage. Then similarly, we had lots of amines, alpha amino acids. Alpha amino acids join together to form proteins. What is that bond called?
Polypeptide.
Polypeptide. Similarly, the nucleotides are going to join together and bond form. Proteins amino acids alpha amino acids we need to understand what is a nucleotide first, and then how they are grouped together. Okay, so that is what is going to be our approach here. Now, so if I had, let's say I have a DNA or a RNA, okay, then I do complete hydrolysis. Okay. I do complete hydrolysis. Then I see that the DNA and RNA is basically made up of three things. One, it is made up of something known as pentose sugar. Okay. And then it also has phosphoric acid. Okay. And then it also has nitrogenous. Yes. Okay, so in the moon components, these are the components of DNA, RNA. So which means that first point, the nucleic acid or DNA, RNA is basically a polymer of nucleotides. I am also giving you something else. So which means that nucleotides is something related to all these three only. Okay, so that is what we are going DNA and RNA, we are looking at two things. So first one, let's talk about pentose sugar. What is pentose sugar here? When I look at the pentose sugar, I have two things for DNA and RNA. If I'm looking at my DNA, the pentose sugar I have is beta D2 deoxyribose. That is my pentose sugar. And when I look at RNA, my pentose sugar is basically beta D ribose. And what is sugar? Basically, sugar is
Carbohydrate.
it is a carbohydrate, saccharides. Ribose, maltose. So when I say pentose sugar, it is basically a carbohydrate. So, but the carbohydrate that is present in the form of pentose sugar for DNA and RNA is different. For DNA, it is beta D2 deoxyribose, whereas in the case of RNA, it is beta D ribose. So, how does this look from a structure wise? So, let's take first beta D ribose papa. So, when I look at beta D ribose, it is a pentose sugar. Okay, so it is like a 5-ring structure. Okay. Now I have CH2OH at the top. Then I have H. Here I have H. Here I have OH. Here I have H. Here I have OH. Now, if you recall, the beta on the beta on the what is the difference between beta and alpha?
First, second carbon வந்து
So beta la in the two heavy groups are opposite to each other, correct?
yes.
Is opposite to each other, right? The same thing is going to be here. So I will have OH opposite to each other. So that is what is the thing with beta. So this is beta D ribose. Now, and let's say this is my first carbon, second carbon, third carbon, fourth carbon, fifth carbon, pentose sugar, so 5 carbons. Now, what is beta D2 deoxyribose? So it is the same thing. It is a ribose only. It is a beta. It is a D. But second la வந்து deoxy. So the second la இருக்கிற இந்த OH la இந்த oxygen கிடையாது. H மட்டும் தான் இருக்கும். அவ்வளவுதான் difference. Okay. That is what is beta D2 deoxyribose means. I can draw the same thing. So it will be CH2OH. This is H. This is H. OH. This is H. This is OH. H. Inga, this oxygen is not there. Okay. That is the only difference between beta D ribose and beta D2 deoxyribose. Okay. So that is what is my pentose sugar. Now, next is, let's look at the nitrogenous base. Nitrogenous base. Okay. So nitrogenous base அப்படின்னு சொன்னா என்னது? So it is some nitrogen containing. Okay. So we generally say that this nitrogenous base, it is a derivative. It's derivatives of purine and pyrimidine. Okay. இதெல்லாம் கொஞ்சம் nitrogen containing compounds. We're not going to go into detail of these things. Okay. Now, in the purine, right? Purine base or the derivatives are two components. One is adenine, which is represented as A. The second one is guanine, which is represented as G. Okay. Then in the pyrimidine base, we have
Cytosine.
three things. One is cytosine, which is given as C. Those structures, don't need to worry. And then you have thymine, which is T. Okay. Then you also have something known as uracil, which is U. Okay. Now, these are the nitrogenous bases. Now, if I look at the DNA, the nitrogenous bases that are applicable are A, G, C, and T. Okay. If I look at RNA, the nitrogenous bases are A, G, C, and U. In the thymine வந்து RNA ல கிடையாது. Okay. Whereas DNA ல thymine இருக்கு. So these two are different. This thymine and uracil is different. The other three, adenine, guanine, cytosine, are common for both DNA and RNA. Clear? Now this is separate. இப்போ nitrogenous base னு தனியா பார்த்துட்டோம். Pentose sugar னு ஒன்னு தனியா பார்த்தோம். இப்போ என்ன போகுதுன்னா, ஓகே, இந்த nitrogenous base இருக்கு இல்லையா? This nitrogenous base, whatever I have here, when this nitrogenous base is going to be present in the C1 carbon of this pentose sugar, then it is something known as nucleoside. Okay. So, nucleoside. The nucleoside is where my base is attached to the C1 of my pentose sugar. So, whatever pentose sugar I have, whether it is deoxyribose or ribose, that doesn't matter. Okay. First, second, third, fourth. So, I had CH2OH, I had H, I had H, I had OH, I had H, I had OH, I had H. In the OH, I'm going to have a base here. Now, this base could be anything. This base could be adenine, guanine, cytosine, thymine, uracil. I mean, uracil irundhuchna RNA ku inga irukkalam. Whereas for DNA, thymine irukum. Okay, so uracil cannot be there. Are you clear till now?
Yes.
Okay, so nucleoside is basically my pentose sugar plus nitrogen base. So, இப்போ வந்து ரெண்டு வந்துருச்சு பார்த்தோம். So, we know pentose sugar, nitrogenous base. இது ரெண்டும் சேர்ந்துனாச்சு nucleoside ஆச்சு. இப்போ அந்த nucleosideல phosphoric acid add ஆச்சுன்னா it becomes a nucleotide. அவ்வளவுதான். Okay. So, what is pentose sugar we saw separately, nitrogenous base we saw separately. Now we mixed these two, we call it as a nucleoside. And in that nucleoside, now phosphoric acid is going to add, then it becomes a nucleotide. Then all nucleotides join together to form a nucleic acid. This is how you kind of relate it. Okay. Now, let's look at nucleotide. When the nucleotide, what is nucleotide? I had my pentose sugar. Adoda, I have the base at C1 position, correct? Now I'm also going to have phosphoric acid at C5 position. You remember this, then you can draw the nucleotide. But C5 another, which was my C5 here? 1, 2, 3, 4, 5. Upper and the fifth idula, I'm going to have phosphoric acid. So what is phosphoric acid? What is phosphoric acid? H3. H3.
3PO3.
That is your phosphoric acid. Now in the phosphoric acid, it has to react with this CH2OH, right? And it needs to be kind of formed. So what is going to happen here is, let me write it here. The phosphoric acid is H3P O4. So this is my phosphoric acid. If I have இங்க என்ன இருக்கு CH2OH இருக்கா? So I have, I'm going to write like this, CH2OH. Now what will happen is if they add, water will go away, right? Sorry, okay. Okay. The water position now. What will I have? I will have something like OH, P, double bond O, OH, O, H2, C. Clear?
Yes.
Okay. Now this is what will get added in my fifth position. So if I draw the diagram for nucleotide, it will be like this. Okay. So in I have my base, right? And then I have my H, have H here, have OH here, I have H, OH, have H. I will have CH2, O, P, double bond O, OH, OH. Now what is this? This is nucleotide. Prida.
Yes.
The nucleotide la basically you have what? Pentose sugar plus nitrogen base plus phosphoric acid in this. So sometimes on the book la OH kudukama, they might have just given O minus everything kuda kuduthirupanga. Anything is fine because it's an acid, right? So if you represent pannirkom sila book la, you can also write OH. So both are right. Now this is nucleotide. So now what is DNA RNA? It is a DNA RNA is a polynucleotide. So which means that these units need to join together. Now the question comes, எப்படி join ஆகுது, right? எப்படி join ஆகுது, எந்த carbon join ஆகுது, and what is that linkage called? Okay, so that is the important one. So when I say nucleotide, okay, so the carbon of the fifth carbon plus third carbon of two nucleotides will join together. Okay, and what is it called? That linkage is known as phosphodiester linkage. Okay, so the important one. So எப்படி join ஆகுது? The phosphodiester linkage. Okay, so let's see how it goes. This. So I have a base, I have H, I have H, I have OH, then I have H, I have OH, then I have H. Here I have my CH2, O, P, double bond O, O minus, O minus. Okay. Then space, this is H, H, OH, H, H, this is H, this is C, H2, P, double bond O, OH, OH, P, double bond O, OH, OH. This is 1, 2, 3, 4, 5. So, the fifth carbon and third carbon. So, which is these two. So, here I have, let's say, my OH. You join Anna, H2 available. Is it here?
Yes.
Okay. Then what will happen? This is your base, H, OH, H, H. And here I have my H, CH2, O, P double bond O, OH, OH. Now this is linking to oxygen, right? And oxygen level or phosphorus, this is O, H2C, double bond O, OH. Now clear. So this one is known as your phosphodiester linkage. Can you tell me why it is called diester?
எனக்கு வந்து ரெண்டு POR group.
Huh?
POC
Hmm.
வந்து ரெண்டு இருக்கு and there is P double bond group.
அதனால diester. What happens when alcohol plus acid reacts?
Ester.
This gives ester. Alcohol and acid எங்க react ஆச்சு first? இங்கே react ஆச்சா alcohol and acid? This is alcohol, this is acid.
Yes.
இப்போ இங்கே ஒரு esterification நடந்திருக்கா நியூக்ளியோசைட்ல நியூக்ளியோடைட்ல?
Yes.
Yes.
அப்ப இங்க திருப்பி இன்னொரு acid and alcohol. So this is OH is your alcohol, this is your இன்னொரு acid. So that is why it is known, and it is linked via the phosphorus. So that is why it is known as phosphodiester linkage. So this is what is the structure for phosphodiester linkage. Very, very important. So nucleic acid la kandipa or question varalam. So we get it just if you draw like this, then it is fine. So but enna pandrona, for various cases it is not possible for us to kind of draw all these things. So simple representation is we just say sugar. The sugar la or base irukku. Then I have a phosphate linkage. Then I have a sugar. And then I have a base. Then I have a phosphate linkage and so on. So in the Mari, this is a simple way of representing. So this thing, right? When I say sugar and phosphate, I can just say represent n times. Right? So this can be another sugar, another base, and so on.
Sir, இங்க வந்து இந்த மேல இருக்கிற டயக்ராம்ல வந்து POH bond இருக்குல்ல, அது வந்து இன்னொரு P and OH. அது வந்து இன்னொரு இதுக்கு லிங்க் ஆகும். Will it link to another?
இல்ல இந்த இது வந்து இன்னொரு இதுக்கு போகும். இப்ப இது வந்து இதுக்கு லிங்க் ஆயிடுச்சா? இதுல 3 இருக்கு இல்லையா? This is the third one, right?
Yes.
So this third one will link with the another fifth one of another nucleotide and so on.
அப்ப வந்து ஒரு phosphorus will always have one OH attached to it.
ஆமா.
Okay.
இந்த இது this whole thing is your diester. இது அப்படியே இருக்கும்.
Okay.
Okay. So that is what is nucleic acid. So let me quickly revise. So we saw that nucleic acid is nothing but a long chain of nucleotides. And இது ரெண்டு டைப்ல இருக்கு, DNA, RNA. So nucleotides in sona, it is composed of pentose sugar plus phosphoric acid plus nitrogen base. So pentose sugar lada, we have this difference between these two, and we say that it is nothing but sugar and sona saccharides, right? It is one of your carbohydrate. So it is a kind of a furan structure. A 5-ring structure is your furan structure. I'm already a 5-ring la paathirkom. So adeda ingiyum. So beta D ribose is this, and the second idula oxygen poidichna, it is beta D2 deoxy ribose, and then you have these 5 nitrogenous bases, but anytime only 4 is applicable. So DNA la vandu AGCT are applicable, RNA la vandu AGCU are applicable. Okay, then nucleoside is nothing but one of the base is attached to the first carbon of the pentose sugar, then it is called as a nucleoside. Okay, then on the first on the nucleoside la fifth carbon la phosphoric acid attach aayirudhuchna, then it becomes nucleotide. Okay, then multinucleotides or polynucleotides, right, when they join together via the fifth carbon and third carbon via what is known as a diester linkage, then it becomes a DNA or RNA. So இந்த இடத்துல வந்து இப்ப பேஸ் மாறிச்சுனா it could be anything. DNA ல இருக்கிற 4 இதுல வந்து depends on the base. Sorry, DNA ல வந்து இந்த OH இருக்காது. So RNA ல இதெல்லாம் இருக்கும். Base will be different. There can be 2 types of bases. DNA ல அதுக்கு உண்டான corresponding bases இருக்கும். You will not have this oxygen here. So that is your general structure. Okay. So if they ask you to write, especially giving you a base, போட்டு இந்த மாதிரி ஒரு ஸ்ட்ரக்சர் போடுங்கன்னா, you need to be very careful here. இங்க OH போடக்கூடாது for an RNA. DNA க்கு தான் இங்க OH போடணும். And whatever is the base applicable, DNA க்கு deoxy, so there will be no OH. For RNA, you need to put. Okay.